Good evening, everyone, and welcome to the virtual international conference on global research trends in biotechnology, hosted by the Department of Biotechnology from St. Joseph's College of Engineering. Before we start, like the previous session, I request you to follow a few rules to ensure the smooth sailing of the event. Please do not unmute your microphones throughout the session and do not introduce yourselves in the chat box. Kindly post any questions you have in the chat box and they will be answered at the end. If you face any technical difficulties at any points, relate them in the chat box and we will look into it. I suggest you all to use your microphones for better quality, and the feedback form will be posted in the chat box anytime during the session. I thank you in advance for your cooperation. Now let's get into this evening's session. Our speaker for this evening session is Dr. Jovvian George Parakkal. He completed his bachelor's degree in industrial biotechnology from St. Joseph's College of Engineering in the year of 2007 and went on to obtain his master's degree from Rajalakshmi Engineering College. He then pursued his doctorate from the National Institute of Research in Tuberculosis at ICMR NIH International Center for Excellence with his thesis on the influence of element infections on the immune response to latent and active tuberculosis. Dr. Parakkal has published over 20 papers in several renowned journals, including the European Journal of Immunology and Journal of Infectious Diseases. He is currently a senior research fellow at the Laboratory of Molecular Parasitology in the New York Blood Center and is working on creating a potentially superior viral vaccine by using activation-associated secreted proteins from the filarial nematode Onchocerca volvulus as an adjuvant with the trivalent influenza vaccine and the Middle East Respiratory Syndrome RBD. Once again, it is an immense pleasure to have you with us today, sir. I request you to take over.
Thank you so much, Swatika, for the nice introduction. All right, so here we go. All right, yeah. So thank you so much again. So I'm Jovvian. I work at the New York Blood Center, and I specifically work on helminth infections. And of late, we were kind of trying to create a vaccine for helminth, especially the Onchocerca volvulus that Swatika was telling you. And in the meanwhile, we were also able to find out certain proteins that were able to act as adjuvants, meaning they are immunogenic, they're able to stimulate human cells to secrete certain cytokines and in turn increase or enhance antibody production. So these are classic qualities of how adjuvants work, and so therefore we try to use a part of the protein from the worms as adjuvants in various viral vaccines. And for today's talk, I'll be mostly focusing on the mechanism of one such adjuvant, which is ASP1, using influenza vaccine as a vaccine model. And if time permits, I can try to show how it works even in a coronavirus vaccine model as well. So I come from New York Blood Center, so it has nothing to do with filariasis or vaccination. It is a global blood donation kind of a company, and they are a conglomerate of various cord blood programs, and their main aim is to cater experiments or helpful in cellular and transfusion products, especially for patients who are needing various blood and its blood products like sickle cell anemia, malaria, dengue, chikungunya, and those kinds of diseases. And we are also a company that are able to create detection assays, detection tests for various infectious diseases. So on the whole part of New York Blood Center, we are a small group of laboratory research in molecular parasitology. And our lab mostly focuses on trying to find out how to minimize viral infections, especially the pandemic strains. So over the past decade, we have been exposed to not just one, but 4 other pandemic infections. So it all started with SARS in 2003. So I'm starting with SARS because most of you were alive to see or experience this pandemic. So before SARS, the most deadliest pandemic was, of course, the HIV/AIDS. So in 2003, there was the SARS outbreak, and the number of casualties were 500 globally. And then in 2009, you had the swine flu, which had the highest so far before the COVID-19 was there. with 100,000 cases globally. And then you had in 2012, which is MERS. So MERS and SARS are both the coronavirus family. So even the MERS is called Middle East Respiratory Syndrome. It had an outbreak in Saudi Arabia and North Korea, and they had again at least 700 cases casualty globally. And then there was Ebola in the year 2014 to 16, which had a roughly of 2,000 fatalities globally. And then, of course, we have the most recent COVID-19, which has now crossed what influenza had acquired. So it is like around 350,000 casualties globally, and unfortunately, it's still counting. So our lab is basically interested in trying to find vaccines for the most pandemic strain out there. So over the course, I'll be especially focusing on the influenza swine flu. Why? Because in the US, people are still infected with the influenza infection. And the problem with influenza, unlike coronaviruses, is that they mutate very, very quickly. So before I could move on to some basic data and about vaccines and all, I would just like to take some time in introducing what influenza virus is and how different is it from other viruses. So the influenza virus has basically 5 proteins. So the most important ones are the heme agglutinin, which is called as HA, and the Na, which is the neuraminidase. So these are the proteins that are found at the outside surface of the virus, and the HA especially are the ones that recognizes your sialic acid sequence on the human host cell. So without the sialic acid, the virus cannot bind to a human host cell and infect it. So once it recognizes a sialic acid receptor on the human host cell, it creates an endosome, and then it enters into your cell. It releases its genetic material. It hijacks your RNA and DNA machinery, and as in when your own cell replicates, the virus also replicates, and then in the form. Give me a moment. Yeah. So what happens is, and then it packages itself out, and then it buds out of the cell and moves on to infect another healthy cell. And as and when this happens, you have multiplication of the virus itself and multiplication of infected cells, and your own body will not be able to clear the infected cells. So that is how a virus infection happens. So people have tried to use vaccines to eliminate this kind of an infection. So far they have isolated the HA part of the virus, and they have created vaccines for it. So the HA, this part is particularly called the HA, the hemagglutinin part. So it has two subsets. So you have one which is the globular head, and then you have a linear stalk. So it is based on this that vaccines have been created. So they isolate the HA part of the vaccine, the virus alone, and they're creating vaccines out of it. So hence the vaccines are not live, or they are not inactivated. So they don't have any viral genetic particle. This is just a protein coming from the outside part of the virus. So in any case, even having you vaccinated with this is not going to kill you. However, what happened? Yeah. So they thought it was an easy step, but the problem what we face with influenza every year is something called the antigen shift or strain reassortment. So it means that every year you're having H1N1, or you're having H1N2, or you're having H3N2. The strain differs every year in the atmosphere. So therefore, there is no one single monovalent vaccine that can be given to a person to get himself protected. So it is believed that the H1N1 is actually a swine flu, meaning it is coming, the source is from a pig. However, research have stated that the initial origin of H1N1 was actually from birds. So it was basically an avian flu, and it had transmitted to pigs, and it mutated in pigs, and it started infecting pigs. And then from pigs, it came into contact with humans, and it went on, and humans became a good host for the virus. And then what happened next was there are so many strains that mutated in the pig from H1N1, and then you had H3N2. And then you have something called a mixing vessel, where you have a strain of H1N1 coming from humans and a strain of H3N2 coming from pigs. And they both mutate in a pig, and it comes out into a new strain called H1N2. So, and now what happens is you have a vaccine for H1N1, but you do not have a vaccine for H1N2 or H3N2 if there's an outbreak. So this is called antigenic shift or strain reassortment. So that is why every year you see people, even though they are getting vaccinated, they still get infected with the influenza infection. It's because the strain what is prepared in the vaccine is not similar to what is circulating in the atmosphere. So that's one of the challenges what we are facing with influenza vaccines currently. So therefore, companies have created something called the trivalent or quadrivalent vaccines so that they actually isolate the HA proteins from various strains of H1. So they have isolated, in this example, is a trivalent influenza vaccine. So they have the HA part of one of the strains from H1N1, and they have an HA of H3N2, and they have an HA of influenza B vaccine, influenza B virus. So the problem here is the HA is a protein. It is not, as I said earlier, it is not live, it is not attenuated. Therefore, it's not so potent to actually protect 100% of the population. So that's where adjuvants come into play. So what are adjuvants? So adjuvants are molecules, can be protein, can be enzyme, can be a viscous material, and their sole property is to enhance whatever the vaccine induces. So these proteins are very poor inducers of antibody pure inducers of antibody production. Therefore, adjuvants help in enhancing this particular antibodies against the vaccine so that you build more and robust neutralizing antibody titers in case of for protection. So one of the arguments that we were kind of interested was the ASP1. So we were in the middle of trying to find out a vaccine for Onchocerciasis volvulus, so which is a helminth nematode worm, very, very similar to the filarial worm what you see in India, which affects mostly people from the southern and the northeastern states. So it's called Ucheria branchofti abruzzi malai. So, and commonly they are people who get infected with such worms are normally termed as people who have elephantiasis, Yana coil noin solvaga. So it is very, very similar. They both are acting very similarly. So they are transmitted into the humans. So in the case of volvulus, it is through a black fly, but in the case of filariasis, it's through mosquitoes. And these worms enter into your body. They kind of form a niche, and a nodule is created, and they lay eggs or they make babies in these niche, and that's how these people get infected. So the idea was to isolate these worms from these people, and then try to isolate certain components from the worms and try to create a vaccine against the worm. So we came around, we narrowed down to like 5 candidates which were able to show some signs of protection from the vaccine that help, meaning that it helps to not make the larvae migrate from L3 to L4 stage. So one such protein was the ASP1, though it did not help very much in filarial vaccine concept, but we were able to find that it was able to induce antibodies on its own. And when we stimulated cells with ASP1, we were able to see that these cells were making IL-10 gamma and IL-10 cytokines. So these are classical signatures of something which is immunogenic and something which can enhance a vaccine component. So this is where ASP1 is. So we tested this ASP1 on various viral candidates, especially HIV1, the SARS coronavirus peptide back in 2003. And for now, we were now able to test this on influenza vaccine, and it was giving some good results. And therefore, I thought, why not share how this actually enhances a very common vaccine in the US, which are not able to protect 100% of the people over here. So before moving on to the mechanism of action or my data, I would like to focus in this particular slide because this is going to be repetitive so that you kind of understand what goes into finding out how a mechanism of a particular molecule is reacting in a host system. So in a normal scenario, when you have a vaccine or a vaccine with or without an adjuvant is immunized intramuscular injection. So you get your vaccine shot on your muscle, which is probably your biceps. And what happens is your body senses these foreign molecules, and it passes on to two different immunity. So one is the innate immunity, which are comprising of the innate cells, which are nothing but monocytes, macrophages, and DCs. So, and then it passes on to the adaptive immunity, which is nothing but comprised of adaptive immune cells, which are nothing but T cells, your B cells, and NK cells. So, for once you have a vaccine immunized intramuscularly, what happens is these molecules disintegrate into your muscle tissue. And in the muscles, you're able to see the first form of defense cells that come into action are your monocytes, macrophages, and DCs. So, these are your innate cells. And their role is to identify those molecules using receptors or pathogen recognition receptors. So you have many of them. You have TLRs, you have RLRs, you have NLR, which are inside the cell, and you have certain receptors which are on the surface. And the way of clearing infection is by two concepts. One is on its own using phagocytosis method, where it eats up and digests the material on its own and clears the infection. If it cannot do it on its own, it is going to need help with adaptive cells, where they again digest and they present them to T cells using the MHC class II molecule. So this is where a transition happens from innate cells to adaptive cells. This is called antigen presentation. So now here with T cells, the main functions of the adaptive immunity is about T cells helping your B cells to make antibodies. So that is a very, very common aspect in vaccine mechanism. So you have your T cells in there. So T cells will secrete certain cytokines that tell your B cells to produce antibodies, especially neutralizing antibodies, which then neutralizes your pathogen. Whereas the T cells that help the B cells to do that are TFH or T follicular helper cells, or CD4 positive T cells, which are nothing but T helper cells. So the main function is to help B cells secrete or produce IgGs, IgMs, and IgAs and Ds. So the ones that are important in neutralizing a pathogen is the IgG. And then you have other cells that directly interact with pathogen and kill them via secretion of interferon gamma or perforin on granzyme B. So those cells are actually called as cytotoxic T cells, which are CD8 or otherwise known as CD8 positive T cells. And most of them can also be NK cells. So this is how the mechanism of a vaccine with or without an adjuvant actually works. So the adjuvant just multifolds this aspect of a vaccine. So with this, we kind of have an idea of how it works with the influenza vaccine model. So this data was published before I joined the lab. So basically what we kind of did was, my peers, what they did is they immunized mice with mock, that is they just with PBS with saline, and they immunized the mice with the vaccine, the influenza vaccine, and they immunized mice with the vaccine with our adjuvant. And then it takes at least 14 days for your mice to induce or produce those IgGs by your B cells by day 14. So at day 14, we're going to collect blood and look at their antibody responses towards the vaccine in the blood by day 14. And we look at their antibody responses, and then by day 21, we challenge them with a mouse adapted H1N1 strain, so which is nothing but PR8. So and then we look at their weight loss to see if they're losing weight. So if they're feeling sick, they end up losing weight, and then that's how we determine whether they will survive the infection or not. So if you look at the black bars for the moment, so we have the first black bar on the left you will see is we've given a 3 microgram dose of the vaccine alone, and we're given a 3 microgram dose of the vaccine plus our adjuvant, and you're able to see clearly that the vaccine with the adjuvant is giving you more antibody titers than the vaccine itself. So this is from young mice. So in reality, people who are senior citizens and people who have comorbidities are very susceptible or vulnerable to influenza infection. And you've very well heard this time and again with the COVID-19 as well. So we kind of did an experiment where we used aged mice. So these are mice which are 78 to 82 weeks old mice. So they're practically, when you do an age match with humans, they're actually they're similar to people who are above 65 years of age. So even in those age mice, we were able to see that the vaccine with the ASP1 adjuvant were giving heightened antibody titers compared with the vaccine alone. And the other property of adjuvant is to reduce the dose of the vaccine itself. So we went on from 3 microgram dose, we went down to zero point 0.6. So that is kind of 9 times lower. So when we were able to reduce the dose of the vaccine alone, so we used from 3 microgram, we used 0.6 microgram, and then we used with the adjuvant, and we were able to get a heightened antibody responses compared to the vaccine alone. And we used alum as one of the positive controls. And alum is nothing but an aluminum hydroxide or aluminum phosphate adjuvant, which is the only adjuvant at that time which was approved by the FDA, and it's been used for many different human vaccines. So this was interesting data, and they were able to show that the adjuvant was able to enhance antibody production. And we looked at IgGs. So IgGs have subsets. So they are IgG1 and IgG2. So IgG2Cs are the one which are showing more effects of neutralization, probably. So we looked at the IgG1 data and IgG2C from the total IgGs, and we were able to see that the TIV plus the ASP1 vaccine was able to induce more IgG2C than what the vaccine alone induces. So this was classic, which was new, and it was outstanding at that time. And when we looked at the percentage survival, we were able to see that the mice that received no vaccine at all, or the mice that received only the vaccine alone, had only approximately 25% of them only survived the H1N1 challenge, whereas the mice that had the vaccine with the adjuvants, whether the ASP1 or alum, they had at least 83% chance of survival. So this was a data, and my boss, I just joined the lab, and my seniors or my peers were having a tough time getting this published because this concept was something new. No one knew how it works, but we somehow our lab kind of rebattled so many reviewer questions saying that okay, because this is novel adjuvant and no one is buying uh, our research. So they were the very common instances what the critics were asking was, how is this possible? What is the mechanism? And how is this possible? What is the mechanism? So at that time, there was no immunologist in the field, in the lab. So I was recruited for this project. And then I had a very eventful journey of trying to find out the mechanism of this particular adjuvant at that time. So what we understood from the previous data was you are able to reduce the dose of the vaccine. So as well, why not we reduce the dose of the adjuvant itself? So initially in 2016, they were using 25 microgram of the adjuvant, and they were using 0.6 and 3 microgram of the vaccine. So I told them that let's reduce the dose of the adjuvant and see whether it improves the antibody responses, and can it protect the mice 100%. So if you looked at the previous data, you're able to see a protection of only 83%. So, and then if you look at these figures, you reduced it 9 times, you had better antibody responses than with 3 microgram ASP. So we decided why not go down. Let's do a more dose-dependent curve to see whether it actually improves antibody production and therefore improves protection. So what we were able to see is we reduced the dose of the adjuvant from 25 microgram to 12.5 microgram, and we were still able to see that it was able to induce enhanced IgG production compared to the vaccine alone. And we decided to go down further. So from 0.9, we went down to 0.2 to 5 microgram of the dose of the vaccine alone. So with 0.2 to 5 of the vaccine and 12.5 of your ASP1, you were still able to have better antibody responses compared to the vaccine alone. And surprisingly, we were able to see that in both cases, the one in red, the vaccine plus the adjuvant, all of the mice survived. So we've used like 5 mice per group in this study, 3 to 5 mice, and we were able to see all of them survive. So that means it is 100% protection. So I know this would be too much for you people, but this is how vaccines and adjuvants work. So in layman's term, what I'm trying to say is that we had a vaccine which we were giving 9 microgram for a mice to actually make sure that it survives a H1N1 infection. So now what we're doing is we've reduced the dose of the vaccine from 9 microgram to 0.225 microgram. So that is like 40 times lowering the dose. So in reality, you have a vaccine production and you end up getting a pandemic and you don't have sufficient vaccine to give to 1.2 billion people in India. And they have produced only 100 million vaccines and say you don't have much. So all we have to do is just procure vaccines for 30 million people and then add our adjuvant, and it is going to be enough for 1.2 billion people. So that is the power of adjuvant, and that is what we were showing in this paper. And again, we had a tough time uh, selling this concept out, and it was we were given very harsh critics to say how is this possible, blah blah blah. So we wanted to find out the mechanism of how our adjuvants actually helped in enhancing a common influenza vaccine. So what we understand from this picture again is we wanted to go from backwards to top. So we understood that you definitely need antibodies for protection, and those antibodies are coming with the help of CD4 T cells. So we want to know whether is this the pathway or is it the cytotoxic T cell pathway that is involved in protection based with our adjuvant. So what we did is we procured mice which were deficient of CD4 positive T cells and mice which are deficient of AID, which is an enzyme that converts your IgM antibodies to IgG. So what we were able to see is in a wild type mice, that is in a normal inbred mice, you are able to have an enhanced IgG production. However, when the mice which lack the CD4 T cells and lack the enzyme that converts IgM to IgG, they were not able to produce any antibodies, suggesting that the ASP adjuvant is required. I mean, the CD4s and the B cells are required, antibodies are required for protection, especially with the adjuvant itself. So what we're looking here is weight loss after challenge with the H1N1 infection. So if you look at the black line, so those are the mice that were given the vaccine plus our ASP1 adjuvant. So you are able to see that all of the mice succumbed to the H1N1 infection. It means that it requires CD4 and the AID enzyme to actually induce protection. So now we found out what is needed to protect. So you need B cells that produces antibodies, and you need the T cells that actually help in B cell to produce antibodies. So the next is we moved on to the innate cell compartment to see what is the particle, what is the mechanism. So it was easier to choose, but the problem was if you look at these data. So financially, what we were facing was a wild type mice, a normal mouse will cost $30, and a deficient mouse like these will cost me $300. So my boss was like, and she was very generous enough for me to use 7 mice per group, and that was a lot of money. So she was like, I mean, we'll have to, put in a plan together to see which ones to choose, which knockout mice that we need to choose to identifying what the innate cell, what cells are important in, or what molecules are important in the innate cell signaling. So what we did is we used a technique where, which normally every people use, is to first try it on in a dish rather than trying it on animals. So we did something called PBMC isolation. So I hope you people know what PBMCs are. So they are polymorphic mononuclear cells, or peripheral blood mononuclear cells. So these are cells that are on your surface when you are separating it from a Parakkal gradient manner. So you have blood, you layer the blood on your Parakkal, which is a viscous material, and then you spin them down using centrifugal force. You're going to see separation of different cells based on their weight or granularity. So your cells, especially your T cells, B cells, and monocytes, will be forming a ring which is called a buffy coat, and below the ring are your RBCs and neutrophils, basophils and eosinophils. So in this buffy coat, we were interested in isolating the monocytes And we stimulate these monocytes, or we enrich these monocytes by adding GM-CSF and IL-4 cytokines. So we program these monocytes to become dendritic cells. So once we have these dendritic cells, we then stimulate them with a control protein or with our adjuvant, and then they become activated or not. So we need to know whether they get activated, and once they are activated, we check them using flow cytometry, and then once they are activated, we isolate the RNA, and then we send them to do an RNA-seq analysis with this. So what an RNA-seq analysis does is it kind of spits out various genes that are associated with a particular pathway. So what we were able to see is it was able to upregulate certain pathways which are in yellow, and there are certain pathways that are downregulated, which are in blue. So in most cases, we are able to see is we just chose the top 15 or top 10 pathways. We wanted to see what are the top 10 pathways that the ASP is able to activate. And there were several, and these are all mostly all the common ones what an adjuvant does. So, but what we found interesting was we were able to find a hit called toll-like receptors, and then we had a hit called interferon signaling, and we had a hit called IRF. So this pattern, I will let you know later how we came into a conclusion to what to start with or what to start with. But the icing of the cake was toll-like receptors. Why? Because you have more than one receptors in your innate cells. So toll-like receptors are the ones which are partially on the surface, and then there are some inside the cell. And you have other receptors like the NLRP3, RLR, and NLR, which have not been activated by ASP1. So we can now focus our interest on TLRs. So a mammalian TLR has more than 10 TLRs, so I think 13 to be in specific. So there are like around 5 to 6 TLRs which are on the surface of your cells, and there are 4 TLRs which are inside the cell, and there is one particular TLR which is neither on the surface nor completely inside the cell. So it took us now we want to decide what molecules, what TLR to choose, which pathway the ASP1 is activating. It was very difficult to choose out of the 13. I cannot spend a lot of money in finding out, in purchasing knockouts of different TLRs and do my studies. So what we were able to identify was out of the 13 TLRs, we were able to see at least 4 or 5 TLRs happen to signal via an adapter molecule called MYD88, and there were 2 TLRs which had a different adapter molecule called the TRIF. So the one in light blue is all the TLRs that go downstream or get activated through the MID88 pathway. So we decided is to now why not try to purchase a mice that is deficient of MID88 and see if the mice survive or mice survive the infection or not. So if it survives, it means MID88 is not involved in protection. If it does not survive, then we know that MID88 is important in protection. So we did the study, we purchased MID88 deficient mice, and we compared that with wild type mice, and we were able to see that in a knockout mice, they were still able to generate antibodies, and they were very much higher compared to the vaccine alone. And we challenged them with the H1N1 infection, and we were able to see that in a wild type mice, the one in black are the vaccine plus ASP1 immunized mice, and we were able to see that all of them were protected. 100% of them were protected from H1N1 infection, and only 25% or 50% of them protected from the vaccine immunization alone. And when we looked at the MID88 deficient mice, we were able to see that there were like 80% of them of the mice which were immunized with the vaccine plus ASP1 survived, and the rest of the mice died which had the vaccine or the mock alone. So meaning that probably the vaccine uses the MID88 pathway, whereas the vaccine plus ASP1 not necessarily uses the MID88 pathway, but something else. So we went back to our RNA-seq data from human disease, and we were able to identify what these genes are. So we pulled up these genes and we fed them into a prediction tool using an ingenuity pathway analysis. It's called IPA. So we are able to fit in all these genes to see whether they fall in a particular fashion, in a particular trend, to see where they are linked to. So we were able to see that the ones which which are highlighted in red are the ones which are activated by the ASP1 protein, and the ones which do not have the color, it means that they were not activated by the ASP1 protein. So what we are able to see clearly is that MID88 has not been activated by by the ASP1 protein, but then there was a signal with a TLR4 over there getting activated. So meaning that there is a TLR signaling activation happening, but it is happening in the absence of MID88. So that made it simple for us, because now we ruled out most of the TLRs and we stand alone with just two TLRs, which is the TLR4, which is somewhere in between the surface and the endosome, and then we have a completely endosomal TLR, which is TLR3, that mediates a downstream signaling via the absence of MID88, or which is with the TRIF alone. So the concept of TLRs is that, I don't know if I have explained it clearly, is these receptors are the ones that recognizes your molecules of the foreign antigen or from your pathogens. So it will decide which adaptive immune response your body has to induce to fight that particular pathogen. So for example, you have bacterial infection. So your TLRs, especially the 4, 2, 1, and 6, will recognize the LPS of your bacteria and try to neutralize the pathogen by a particular secretion of certain certain cytokines by the T cells. Whereas your endosomal TLRs are mostly comprised, or they are mostly detecting RNA material. So in most cases, it is um, a viral RNA. So the adjuvant has no source coming from a viral RNA, so therefore we stuck with TLR4, which can detect different types of molecules, a peptide, a protein, an LPS, or whatnot. So we decided to use knockout mice of TLR4 and the TRIF, and what we were able to see is that they have a reduced antibody production compared to wild type. And when you look at these TLR4 knockout mice, we immunize these mice with the vaccine, and the vaccine with the adjuvant, which is in green. And then we challenge them with the virus, and we record their weight for a period of 14 days. And we're seeing is that all of them are turning out to be sick, and we euthanize them once they reach a particular cutoff. So here you're able to see that in a TLR4 deficient mice, they don't survive the infection. In a TRIF deficient mice, they don't survive an infection. But in a wild type mice, you're able to see a classic survival. And we also did a type 1 interferon receptor signaling, which also again shows that it does not survive infection. So suggesting that suggesting that it is probably via a type 1 interferon signaling manner that it is mediating protection. So why I jumped off to type 1 interferon signaling is because we again pulled up data to show what happens after the TLR4 signaling and the TRIF signaling happens. And we were able to again pull up those genes and then put them inside a prediction tool. And when we were able to show that the interferon signaling was important in this particular ASP1 activation. And then we were able to show you that what was in red are molecules that are coming from an interferon signaling pathway. And downstream of intersignaling pathway are molecules that are very similar to other viral infections. So all these viral infections coming out of your STAT1, 2, and IRF9 are actually molecules that are very good candidates for viral drug targets. So these molecules are the ones that ultimately kill the virus in your lung tissues. So if there is an imminent infection, if your body can fight it out, so these are the final proteins that are released from your body in the lungs to clear viral infections. So we are able to see that the ASP1 protein are able to do exactly that. So that's why we use the interferon signaling receptor knockout mice to show you that these are the ones, the interferon signaling are also important in protection. So overall, what we are able to show from this is a vaccine with our ASP1 adjuvant is able to protect mice in a TLR4 dependent manner, and it activates a type 1 interferon signaling. Therefore, it protects mice from H1N1 infection. So this is what we found so far, and at the moment we are kind of trying to create something called universal flu vaccine, because as I told you in the introduction, the strains change every year. So therefore, it is it is very confusing to create a proper vaccine in each year. So we have a strategy to try to create a universal influenza vaccine by combining the stock of H1N1 and combining the globular head with different strains of influenza, so that we are able to now engineer or tailor make a particular viral vaccine which can produce antibodies in a broad spectrum that can neutralize different strains of influenza vaccine. So this is an ongoing project with a lot of collaboration happening. So this is where the influenza story ends, and if time permits, yeah. So I will talk some on the coronavirus, and I cannot show data on the SARS-CoV-2 because it is still under experimental model. So I will show you what I did with SARS and the MERS coronavirus basically. So as you know very well, the SARS-CoV and the SARS-CoV-2 or the COVID-19 2 has the spike protein, so or the S protein as they call it, and the S protein recognizes the ACE2 receptor on your human host, and that's how it enters into your cell and starts hijacking your replication machinery, and then that's how you get infected. So before I joined the lab, they were working on this SARS-CoV vaccine, and they were able to show that they use RBD as the vaccine model to create. So this is not a vaccine that is available, commercially available. It is still a candidate vaccine. People are still working on it. So they have isolated the RBD, similar to how HA has been isolated from the influenza vaccine. So they have isolated RBD from the S protein of the coronavirus, and they were able to show that when you have the RBD plus adjuvant was able to generate higher or better neutralizing antibody titers compared to RBD alone. And this was done in both mice as well as in non-human primates, which are monkeys. So they're able to see that the RBD alone gives you very, very less neutralizing antibody titers. But when it is given with adjuvants, with ASP1 or with even any other adjuvants, you are seeing a heightened neutralizing antibody response. So I was not working with the SARS one, but I am working with the MERS and the SARS 2 currently. So with the MERS is nothing but the Middle East Respiratory Syndrome. It is also a coronaviral strain. So here what we are able to show is you actually need not one but two adjuvants to actually build that higher neutralizing antibody titers. So here what we did is we combined alum and we combined ASP1 to with the RBD vaccine of MERS, and we were able to show that it was able to enhance neutralizing antibody titers compared to just a single adjuvanted molecule alone, a single adjuvanted vaccine alone. So, because the single adjuvanted vaccine did not induce good titers for protection, so therefore we combined two adjuvants and we were able to show that it was better than a single adjuvanted vaccine. So now with the neutralizing antibody titers, so that not all of them are able to inhibit the binding of your virus to your cell. So we were able to create an in vitro model to show that these neutralizing antibodies, what are elicited by the mice, are able to inhibit the binding of a pseudovirus of MERS to its receptor, which is the HDPP4. So in the laboratory, we were able to show that higher neutralizing antibody titers were able to inhibit at least 60% of the virus to enter or infect a cell via the HDPP4 receptor. So, and then the next is, as you know, that antibodies are important. We looked at something called the T follicular helper cell. So I kind of told this in during the introduction slide. So your TFH cells are the cells, the CD4 positive T cells that help your B cells in releasing antibodies. So using flow cytometry, we were able to identify the TFH cells which are positive for CXCR5 and PD1. So this particular population, you're able to see clearly that the population over here is more than the population what you see in mice that were immunized with just a single adjuvant. So we were able to show that the ASP plus alum adjuvanted vaccine was able to induce more TFH cells than the single adjuvanted vaccines alone. And we are also able to show a correlation that the TFH cells positively correlated with the neutralizing antibody titers. So these cells are in the lymph node. So we were able to show that the CD4 positive T cells which are in the lymph node are able to show correlation with the neutralizing antibody titers which are taken from the blood. A very similar association was also observed with the germinal center B cells. So if there is a germinal center B cell that is formed, it means that for sure your B cells are going to start secreting antibodies. And we are also able to show that they are also heightened when you have both adjuvants in your vaccine. And you are able to see a positive correlation, which the ones in black that are able to show that they positively correlate with neutralizing antibody titers. So here now we are able to show, at least with the MERS vaccine, that you need TFH cells and you need germinal center B cells to induce that particular neutralizing antibody titers in mice. So this is a very recently published paper what we just did. So it now shows clearly that you need TFH cells and B cells and germinal center B cells to induce that particular neutralizing antibody titers. So this is the very aspect what we are studying with the COVID-19 as well. Are we able to see these specialized cells that are important in getting out those neutralizing antibody titers or not? So the more the neutralizing antibody titer, better your chances of protection. So to summarize my talk, so what we are able to show is that the ASP1 adjuvant is very effective in enhancing the potency of influenza vaccine. It is able to reduce the dose of the vaccine by 40 times and still is able to mediate protection from the virus via a TLR4, TRIF, and type 1 interferon signaling dependent manner. With the coronavirus vaccines, we were able to show that it was able to induce a higher percentage of TFH and germinal center B cells in the lymph node that are positively associated with enhancement of neutralizing antibody titers against the MERS-CoV vaccine. So all this data was not possible just by me. It was a collaboration with a lot of people in this study. So I'd like to thank people from my labs. My boss is Sarah Lustigman, and then my previous peer was Sonia Jain, and then current people are Nancy and Shabnam. And I like to thank people from University of Pennsylvania and Drexel who are able to give us the virus and the various talks. And we thank people from Baylor College of Medicine and this group especially. So they are the ones who are creating our recombinant ASP1. So we are collaborating with these guys in getting new SARS-CoV-2 vaccine RBD. And we thank the Laboratory of Virology who helped us in doing the neutralization assays with the MERS coronavirus study. And I thank the animal facility, people at animal facility and flow cytometry, and people at NYU who were helpful in creating these RNA-seq data and Ingenuity pathway analysis figures. And finally, I would like to thank the NIH for the funding. So thank you for your patience, and I am open to questions, critics, doubts, comments. Thank you. Oops.
If any of you have questions right now, you can ask sir right now.
Hello, sir.
Yeah, tell me.
It's an excellent informative talk, sir. I have a doubt. In India, what is the mutant variety of COVID-19? Is there any significant in the virulence?
So far, I mean, there is a mutation in the strain, but it has nothing to do with virulence, unlike with the influenza virus. So the strain in India is different from the strain that is in New York, and the strain in New York is different from the strain what infected in Washington. So yes, the strains are different, but there is no bias in virulence. So irrespective of what the mutation is, it is still as virulent as the original strain.
So the vaccine preparation is not based on the mutant strain?
No, not necessarily, because the mutation did not happen in the S protein. So that's where that's the protein that actually binds to the human cells, and there is no mutation in the at least the RBD part of the S protein. So people are trying to create a vaccine from the RBD part of the S protein. So there are mutations in the S protein, but not in the RBD part of the S protein. So therefore, the virulence is similar, and people are working with the RBD vaccine as a vaccine candidate. So there is no mutation in the RBD.
Okay, sir. Thank you. Thank you, sir.
Thank you. Yeah.
Do bioactive compounds targeting NFKB factors have any effect over COVID or MERS?
Bioactive compounds targeting NF-kappa B?
Yeah, have any effect over COVID?
Have any effect over COVID? Okay. So I have not worked in that pathway yet, so I cannot comment on it. But NF-kappa B is a very common transcription factor, which is necessary for any type of, to neutralize any type of pathogen. So, but NF-kappa B cannot be the only transcription factor that is necessary for protection. It needs, at least since it's a viral infection, it needs IRF3 or 7 or 9, which actually ends up in type 1 interferon signaling. So yeah, NF-kappa B can be important, but is not the only molecule that is important for critical protection. So someone, I think Rajkumar, sent a message. Sir, can we be able to find vaccine for COVID-19 within 2 months? If you are working on that, could you tell me what kind of medicine you will be using against COVID-19? Yeah, you cannot create a vaccine within 2 months. It is going to take a lot of time to get a vaccine out. It has to go through a series of clinical trials. Before that, it has to pass animal trials. So we start with mice first. And then, unfortunately, mice is not a natural host for coronavirus infection, meaning the mice do not have the ACE2 receptors, unlike humans. So you cannot challenge the mice with coronavirus. So you need to use a transgenic mice, or you need to use a higher mammal model mice, mammal models such as gerbils or minks, and those kinds of higher animals. So people are working on that, and they have moved from animals, and now they have to do it on non-human primates, which are monkeys, and then they do clinical trials with humans. So only 3 companies now have ventured into clinical trials, and Moderna is one of them that has moved to phase 3. So in clinical trials, you have phase 1, phase 2, phase 3, and they have now moved into phase 3. So let's hope for the best. So it's not going to come in 2 months. It will take a lot of time. Who else have asked questions?
Sir, we have another question here.
Yeah, go ahead.
Yeah, do adjuvants also increase antibody production in the case of common cold virus?
In the case of common cold virus, I haven't tested, so I don't know.
Okay, we have one more here. What kind of in vitro assay are available to work on COVID drug development?
Yeah, so very similar to what we show with the neutralization assay is to use an in vitro. You use a dish basically with cell lines that can express your ACE2 receptor. And what you do is you mix these cells with a pseudovirus, that is a virus that are only able to bind to the cells but not infect, because you cannot use a live virus, especially a pandemic strain, inside the lab unless you have a biosafety level 3 lab. So what you can do is maybe to check your potential drug is to stimulate your cells with that particular drug and mix them with cell lines that express your ACE2 and see whether it inhibits the binding of the virus to the particular cell. If it inhibits, it's a good potential drug target. If it does not inhibit, it's not a good potential drug target. I mean, the drug is not a potential drug.
So one question, this is Dr. Jayashree. This is related to the question. I was the one asking this question. Is there any other test other than using cell lines?
Is there any other test other than using cell lines?
Because we don't have that facility.
The only other way is to do is in live animals. So, I mean, I would actually start testing it with in vitro techniques first, rather than moving to animals. So that's basically you want to do. So especially when you're trying to identify a potential drug, you need to first work on an in vitro data, a lab in a petri dish. So once you have a data on petri dish, you move to animal models. So that's how it is supposed to work. Yeah, but in case you have more money, then you can actually directly do it on animals. But ideally, yeah, if I were you, I would probably first test the activity on in vitro in a dish. If it works, it works. Again, the disclaimer here is it is not necessary that what works in a dish has to work in an animal. At least you will be able to know what kind of pathways your potential drug is activating from the dish, like how I did. So I was not able to judge what my molecule is able to do. So the only best thing to do is to just take my own blood. So what I used is I used my own blood for the experiment, and I stimulated them, the DCs, with my ASP1, and then you just have to do an RNA-seq analysis or RNAs to, if you know what pathway your drug is actually doing, just do a PCR, a normal qPCR, and it will tell you whether there's a presence or absence of that particular molecule in that particular pathway that your drug or potential drug is actually activating or not. If it activates, then you can very well, with confidence, move on to an animal model to check to see whether it works in such in vivo. That's an animal model to work in an animal. So you challenge the animal with the virus, and you give whatever dose you're supposed to give, and you'll have to see whether it survives the infection or not.
Thank you, sir.
No problem.
Okay, this is the next question, sir.
Yeah.
As per the genetic drifting is considered in coronavirus, how long will it take for the genetic modification to occur?
So it doesn't take quite a lot, but I don't know the exact number. But again, it is not a serious concern regarding coronavirus that there is a genetic modification. So because viruses, one of their main function is to modify. So that's why they've been surviving throughout the years. So the main concern is whether the genetic modification happens at the exact site where it enters or where it recognizes the human cells. So thankfully, it is not in that position yet. So there is nothing to worry, and there is no cause of concern if there is a genetic variation between the coronavirus strain. So that's what someone asked me previously, right? So is there any mutation or anything, a mutant strain that is happening coronavirus? There can be a different mutant, because what we were working on with the New York City people is we were able to identify the strain. What came from New York is coming with what was identified in New York City was the strain that came from Europe, not from China. And the strain that came that had an outbreak in Washington State was not from Europe but was from China. So the strains are different, but the virulence or the effect it causes is similar. So it means that the mutation is happening elsewhere and not at the site of where it is recognizing the human host. So it is not a matter of concern yet.
Okay, so the next question is there any chance the newly synthesized vaccine for COVID-19 will differ for different age groups, since an older person would take more time to develop the immunity?
Yes. So especially, I don't know if they are talking about the Moderna vaccine. So the Moderna vaccine is an mRNA vaccine, and it is not a peptide vaccine. They don't use RBD here. So it's an mRNA vaccine, and they are now clinically testing on people who are between the ages 18 to 55. And they have a subpopulation of group which are above 65 years of age. And of course, when you are concerned with senior citizens, you are going to double your dose. It is not going to be the same dose that you're going to give to a healthy adult. So yes, there is going to be a change. The dose will vary for senior citizens, at least by twofold.
Okay, so the other questions is, studies have reported a possible cure using protease inhibitors through drug repurposing using chloroquine and FDA-approved antiviral drugs and synergism. How far are they effective actually?
So, yeah, I don't know what to say. So each drug has activates its own pathway in the human host cell. And hydroxychloroquine is a drug that is used to inhibit a pathway which is activated by malaria. And if you actually look at, if you know what malaria is, so malaria is a parasite that infects RBCs, and coronavirus is a virus that infects lung epithelial cells. So the source of infection is different, the site of infection is different, and the cells of infection is different. So I do not understand how the same drugs will work. And I don't want to give more detail. So I know it's very contradicting and controversial topic, but I have not tested hydroxychloroquine on my COVID-19 sample, so I cannot comment on that. Yeah, but I am sure that what I'm working on is activating on a particular pathway which has no resemblance with hydroxychloroquine.
Okay, sir. And the next question is it possible to develop a personalized medicine to viral infection?
Medicine, so I don't know, if it is a vaccine, it cannot be personalized. If it is a drug, it can be personalized, not because of the person, but I'm saying it because for comorbidity, so people who are having heart ailments or people who are diabetic, maybe some drugs won't work on them, or some drugs are too harsh for them. So in that case, you will have to find an alternate and a safer method for creating a drug. So this is basically for therapeutic drugs and not vaccines, which are prophylactic. The vaccines are a preventive measure, so it does not reflect, it does not have to be tailor-made for an individual. But when it comes to therapeutic drug, yes, this is one of the concerns. A therapeutic drug cannot be given to everybody because not everyone are same.
Okay, so and the last question for today is, by using tissue engineered models, we can save animals and also more duration for trials of medicine or vaccine to come into the market. Is there any improvement in the testing the vaccines or medicines on tissue engineered models instead of direct testing or trials on animals?
I see an animal lover here. Okay, so we have not reached that place yet. And as I said earlier, a normal mouse will cost me $30, and if I have to work on a knockout mouse, it's going to cost me $300. And if I have to move into tissue engineering and scaffolding, it's going to cost me $3,000. So if I want to have my job, I will work with something far less than $3,000. Yes. So yes, maybe in the course of 20 years, there's going to be a resolution where you're not supposed to test anything on animals. That could happen because over the years, people are getting very sensitive. People are getting very tolerant. So yeah, in 20 years, anything can change. There can be a rule where you're not supposed to use animal model. But for the record, when we apply for grants, the NIH has a portion, a column saying that why do you have to use animal models in your grant? So why don't you use in vitro model data to actually show that whatever your hypothesis works? What is the necessity to use animal models? So that is one of the questions that we need to justify of using animal models in our grant. So in the case of adjuvants, you cannot establish everything on a dish. As I said earlier, whatever results you get on a dish doesn't have to be exactly the same when it is on humans. If that was the case, you would have had all the drugs required to cure cancer, to cure HIV, to cure Ebola. But that's not the case. So yes, so we need animals to show that it protects in vivo. Yeah.
Actually, there are some more questions, sir. So can I ask them? Yeah, okay.
Yeah, sure, go ahead.
Yeah. Will we be able to block the receptor on our cell to which the spike protein of COVID-19 binds to?
Yeah, so that's what neutralizing antibodies are about, right? So you need your body to produce those antibodies that can actually inhibit the binding of the S protein or the RBD of the S protein to the ACE2. So that's what people are working with. You want whether it's a therapeutic drug or a vaccine. The ultimate goal is to inhibit the binding of the S protein or the RBD to the ACE2 receptor, and that's what every vaccine people who are working with the vaccines are trying to do. And yes, if it does that, it means your vaccine is good. And what I'm hearing is, and what I'm seeing from the data, vaccine alone cannot do that. It looks like you need an adjuvant. It cannot be done just by the vaccine alone. One is we immunized mice with the SARS-CoV-2 RBD, just like how we did with the MERS, and it did not give any neutralizing antibody titers. But when we added a combination of adjuvants, it was able to induce a higher level of neutralizing antibody titers. And why vaccine is going to be really important is because we just submitted a paper with human data. So we collected samples from New York City people who were infected with COVID-19 and who are not severe. So they were just quarantined for 14 days, and they might have been given some antiviral drugs to get themselves cured. So after they were cured, we took their blood samples and we looked at their neutralizing antibody titers. And what we were able to see is that these people did not induce very high levels of neutralizing antibody titers, meaning you are not protected from a second wave of infection. And unlike because I'm seeing a lot of news articles, and my parents are also sharing the same thing, is that, so what if you get infected with coronavirus? You get infected again. So that's not it. It is not like chickenpox, where you get infected first, you suffer for 30 days, and then you have lifelong immunity. Unfortunately, it doesn't work with coronavirus that way. So you need a vaccine to boost the neutralizing antibody titer. So for now, what we are seeing is people who recover from COVID-19 have far less neutralizing antibody titers, which is necessary to inhibit binding. So yeah, so this answers at least two questions. One is you need a vaccine to boost that neutralizing antibody titers at least by tenfold. And if there is a second wave, and it is happening in China, in Beijing, you will still be you're not immune to the second infection. So yeah, that's the outcome of this paper, and it is still under review. It is not published yet. Yeah.
Okay, sir, and this is the last question. How about approaching for a vaccine by RNAi mechanism?
How about
approaching for a vaccine by RNAi mechanism?
A protein, is that what you're saying?
I guess so.
Okay, so RNAi is, I don't know how he's linking RNAi with protein. So RNAi is nothing but you're silencing or knocking in an RNA and trying to get a vaccine out. I mean, it is a possibility, but the problem with RNA silencing is that it is not stable. So in the long run. So yeah, I have not tried it, so I cannot comment more than that. I guess it can be a possibility, but I don't know. But it's not stable.
Okay, so thank you for your time. I thank you again for taking the time to join us today and imparting us with your knowledge on adjuvants and its effect on influenza virus, as well as taking your time to answer questions about coronavirus right now. I'd also like to take this time to thank our patrons, our HOD Dr. G. Srikumar, our convener Dr. M. Chamundeshwari ma'am, and our co-convener Ms. K.R. Preeti for giving us the support and making this event possible. And finally, I thank all the participants for your cooperation and ensuring that the event was seamless. We appreciate you for being here. Stay safe, and I hope to see you all in our next session tomorrow. Thank you once again, sir.
Thank you so much, and it was a pleasure giving a talk, especially in my alma mater. And I don't think I've seen any of my previous lecturers here. Or I saw someone say hi. I don't know who. But yeah, I mean, thank you so much. It is a privilege that I was able to give back to where it all started. And I hope this presentation is not necessarily that you need to take home anything. It's just a presentation where I could try my best to invoke some research into people like you. So not many people venture into research of late. So yeah, this is something I cherish, because I like to talk what is happening in the world right now, and the world needs people like you to do research. And thank you so much. Yeah, bye. Everyone have a safe evening.
Yeah, sir. Thank you, sir. Bye.
Bye.